Hi, everybody. Welcome to lecture. And again, I'm your professor, Dawn Murray. And today we are talking about sampling methods. So, what are all the different ecological sampling methods? You have probably experienced some in some of your classes. So, maybe some are new and some are review for you. Many of the sampling methods I'm going to talk about today, you need a permit to do them. So the U.S. Fish and Wildlife Service issues permits for activities that affect native, endangered, and threatened species, migratory birds, and other protected wildlife and plants. The National Marine Fisheries Service, NIMFS, NMFS, but we call it NIMPS. They handle permits related to endangered, threatened, and marine mammal species in aquatic environments. The U.S. Environmental Protection Agency, for example, they manage permit programs for activities that impact air and land and water quality, like discharges into U.S. waters, part of the Clean Water Act. The National Park Service, NPS, they authorize all research and specimen collection in national park boundaries. So, and there's others too, like the U.S. Forest Service, USFS. They issue permits for collecting plants and wildlife on national forest lands. Um, so that's collecting, and you have to adhere to their regulations, reporting back, etc. So before you're going to do any kind of experiments or changing in any habitats, you do have to figure out what permits you need. All right, so how do we collect data as applied ecologists? So there's a lot of methods in the field. These are used to assess species composition. So, what are all the different species that are there, maybe their abundance, how many, and species interactions. And sometimes abundance can look like this, where it's different species and how abundant they are in various plots. There's also lab and genetic methods. So there's a place for that in applied ecology. This kind of studies in a lab might be looking at DNA, species differences, and laboratory and genetic methods often reveal cryptic diversity and relationships among species that you can't observe if you're just looking at them or counting them in the field. With your data, you can also do statistical and modeling analyses. So this is used to quantify diversity, to quantify interactions, to look at dynamics. So that might be inflows and outflows of nutrients or predators and prey, that kind of thing. And then there's also experimental methods. So this is when you manipulate something and you test hypotheses. So a classic example is in the intertidal zone, putting cages out where no sea stars, let's say, can get in and seeing what grows there. Do mussels grow there when they're not being preyed upon, for example? So there's a lot of kind of exclusion type experimental methods that have been really important to tell us about dynamics of predators and prey. The fifth is remote sensing and technological approaches. So this is used for large scale, hard to access communities. So here is kind of some remote sensing. Here's some lab work. Here's some field sampling data. Remote sensing is usually satellites that are collecting data, picking it up, sending it to through the web to computers where then people can analyze the data. This is really, really important. We get a lot of information about winds and currents in the ocean from remote sensing. So here's some of our ways that we kind of look at data. We also want to remember our null and alternative hypotheses. The null hypothesis is always that there's not a significant difference between groups that you're analyzing. And the thought is that any observed differences may be due to chance or sampling error. So, for example, here's a painted bunting, beautiful bird, and an example of a null hypothesis is there's no significant difference between the number of birds at different locations. So the differences that we see in the means of the groups could be due to chance or sampling error. And then the alternative hypothesis is that there is a difference between the groups. So the observed differences between maybe where they are or where they're feeding is most likely not due to chance or sampling error. And so the example there is there's a significant difference between the number of birds at different locations. The difference we see in the means of the groups is most likely not due to chance or sampling error. So let's dive into some of these. Laboratory and genetic methods. And again, you might have some knowledge from reading other papers or other classes. Some of the papers I have you read in this class will also talk about some of these different ways of collecting applied ecological data. So one method is DNA barcoding. So that identifies species from tissue or environmental samples. And that helps you detect cryptic amphibians and insects, maybe things that are harder to see. So that's called DNA barcoding. There's another related environmental DNA or eDNA, and that can detect species presence in water samples, soil samples, air samples, even. So this is a way to monitor fish or amphibian communities. This is done more and more in the ocean. So you can't possibly see everything that's out there. So if you want to know all of the species that use, let's say, the water off our coast, I've worked with some scientists at Stanford in the Palumbi lab, and we've done eDNA monitoring in the Chumash Heritage National Marine Sanctuary. So you go out and you collect water samples. You can also use what we call frammers, which are basically a wiffle ball with some gauze inside that you put on the end of a fishing pole. And you put it out and you reel it in, and you put it out and you reel it in 3 to 5 times, depending on your methodology. You bring it in, and from that gauze, you basically filter it and strain it all out and look at, after a whole bunch of lab work of separating and amplifying DNA, you can see what different species have passed through that water. So fish and plants and sharks and skates and rays, they leave behind, and algae leaves behind mucus. Sharks, urea, whales, poop, fish scales, invertebrates kind of sloughing off tentacles or shedding or molting. All of that can be picked up in these samples of water and the same thing in soil. And you can see the diversity of species. You can't know, there's limitations to each one of these. You can't know, for example, how many there are, but you know that they were there. Another method in this grouping is stable isotope analysis. So this helps you determine trophic relationships and food web structures. So if you have certain, let's say you can measure ratios of naturally occurring stable isotopes like carbon, nitrogen, oxygen, hydrogen, and you can determine geographic origin of a species diet, maybe their migration history of individuals or animals with stable isotope analysis. So it can evaluate the isotopes of elements. Usually it's carbon, um, sometimes nitrogen and oxygen and hydrogen and sulfur, and heavier isotopes like strontium and lead. And those ratios are like a fingerprint for certain materials. So they tell us about the origins of diets and environmental history kind of thing. It's amazing. And then another is microsatellite analysis. Um, Sometimes for short, it's SNP, or microsatellite analysis is also STR. And these are repetitive DNA sequences with varying numbers of repeats. So SNP is actually single nucleotide polymorphism, and it's variations at a single DNA base pair. So you're looking at microsatellite is basically a short repeated sequence of DNA, like 1 to 6 base pairs long, and it varies in the number of repeats among individuals. So it's a high degree of variability. So that's polymorphism, makes them really good ways to distinguish individuals, and you can study closely related populations. So you can use this to study population connectivity, for example, in multi-species forest ecosystem. That's one way people have used it. All right. What about our statistical and modeling approaches? Species richness and diversity, Shannon-Wiener index, Simpson values, evenness indices. These things measure diversity and dominance of species. So this is used if you're like comparing forest patches from one area to another. So there's a lot of theory that go on that's kind of behind these equations, but you can figure these out, and these are numbers based on the number of species and where you are and the amount of area you're looking at. So the Simpsons index, for example, it measures the probability that two individuals randomly selected from a community will belong to the same species. So it's more sensitive to a lack of evenness, and it's influenced by the dominant species. The rank abundance curves, and I don't expect you to know every single one of these. I'm kind of wanting you to understand that there's a lot out there. So when you're thinking about what you want to do for your final project, maybe you want to compare with the species richness equation to different areas, like a shady patch and a sunny patch, or an area near uh, a little wooded area and an area out in the open. It can be something simple like that. I'm bringing all of these up for you to kind of familiarize yourself, but I don't mean for you to memorize like the details of every single one, just to know how many different ways there are for us to analyze data and for us to process questions that we have. All right, a rank abundance curve that helps you visualize species dominance and rarity. So they often look something like this, a really simple kind of curve. And that might be grassland plant community abundance, let's say, or community analysis. So this is an ecological tool, and it displays the relative abundance of species within a community by plotting the species abundance against their rank order. So that representation helps us visualize and compare biodiversity components like species richness, the number of different species. So the richness is the number of different species, and the evenness is how evenly distributed individuals are among species. All right, then we have the species accumulation curves. So that estimates total species richness in a community. That's done a lot with tropical insect surveys, let's say, something that's really abundant and really diverse. So you're plotting the cumulative number of species against a measure of your sampling effort. So how quickly new species are discovered, um, and you're assessing sampling adequacy and you're estimating total species richness. So it's a matter of how much time you're putting in and how much you're seeing. Then there's network analysis with food web. So you're studying trophic interactions and energy flows. So that could be predator-prey relationships in an ecosystem. And then there's multivariate statistics. This is, you've probably heard of principal component analysis or canonical discriminant analysis. These are things I had to use in multiple papers and really kind of powerful techniques to look at dimensionality of your sample when you're comparing a lot of different creatures. Um, so PCA, principal component analysis, is a linear method and it shows you how to maximize variance. Um, and then there's also non-metric, multi-dimensional scaling. So lots of different ways. I don't know how many of you want to be a statistician. It's a great skill to have to know how to analyze various data because labs are always needing people who like doing that, who like doing the kind of different ways of analyzing data and running R. All right, let's talk about some experimental methods. So I gave you the example of an exclusion experiment. So that's when you remove a species and you see how the others respond. So a lot of times you remove like grazers and you study how plants compete when a grazer isn't there. Or sometimes you can remove a predator and see how the prey changes their abundance or growth patterns. Then there's additional experiments where you add a species or a resource to see the effects. So this is done in like that example of the farm in England where you introduce nitrogen. Maybe you introduce a nitrogen fixing plant in a grassland plot and you see if that affects and has more of the other plants around that plot grow faster or bigger, or be healthier than another plot where you don't have the nitrogen fixing plants. And then there's manipulative microcosms or mesocosms. So these are controlled experiments, usually in a lab, with simplified communities. So that's, you could study them in tanks, for example. Maybe it's putting food into certain tanks with different numbers of species and seeing how they compete, how much they compete. Maybe it's having different tanks with different oxygen levels and seeing which oxygen level is kind of the one where they can't survive any longer. That's how we kind of measure a lot of our coral reefs in terms of in the lab, like at what temperature can they not come back anymore. So lots of different ways to do these experimental methods. Let's talk about remote sensing and technological approaches. There's drone surveys and satellite imagery. This is becoming more and more used. You can map vegetation types or you can map habitat structure. This is used in monitoring forests or coral reef communities. Drone surveys are being used for sharks and whales now. And, they can do transects. They can also just follow creatures, see where they're going. So it's like a more used, really interesting way of collecting data. Then there's acoustic monitoring. This is a way to detect species presence and activity through sound. So I had a PhD student who graduated who did this with narwhals in the Arctic. So we deployed underwater acoustic receptors that were basically drilled into the bottom. Lots of permits were needed. They collected sound over years, trying to detect, and there were several of them in various passages, mostly because you probably know that with sea level getting or the ocean getting warmer, and we have less freezing in um, our northern oceans, the Arctic is actually getting more and more boat traffic. And the concern is it's going to affect those marine mammals that really haven't seen the amount of boat traffic that we're getting. And so you can do that to kind of hear the whales. The problem is you have a lot of data to go through. So there's a lot of logging and listening, and we're kind of using some machine learning to have it figure out, like, detect what's an animal versus what's boat noise. I don't know if you've ever heard or listened underwater. I hope you all get a chance to. Boats are incredibly loud underwater. And so just detecting the various whales that are moving, dolphins, there's fish that make sound underwater. So acoustic monitoring is used a lot in the ocean. It's also used a lot on land. So frogs, birds, a lot of sound is recorded and then analyzed to see what is there, what the types of calls are in different seasons, etc. There's also automated camera traps. These are really, really important to record presence, abundance, and behavior, especially of large mammals. So tropical forests like tapirs, um, clouded cats, I think, jaguars, and then in our area, mountain lions and bears. We cannot sit, in a mountain habitat for months and wait for them to walk by in a certain area to know how many there are. So if you put out camera traps that take pictures, if there is motion, that really, really helps us know what's there. Let's look at this example of a common blackbird in Europe. So here we have Spain and we have Northern Europe here. And this is basically showing you an experimental setup with the common blackbird, and it was carrying, they had them carrying radio transmitters, a little backpack right on top, right here, the little antenna. And it was implanted, and they could measure temperature and where the bird was. So they were looking really at seasonal cycles, and they were trying to see where they were in the winter and where they were in the summer and where they were in the spring. So this map shows you, let's see if I can make this bigger. Move it over. Okay, so this they could see in like January, they were wintering at a breeding site. Their average temperature that was cold, right? And then there was also a Southwest population that they were wintering in. They started their spring migration like February, March, and then by April, they were kind of in a warmer area. In July, they were breeding, then they were molting in the fall, and then they made their fall migration back to their wintering breeding site. Here, it shows you their temperature. So wintering is the red areas. Breeding, no, that's down below. The red is the warmer areas here. Yellow is a little less warm, and the blue are the cooler areas. So these little circles show you this Southwest Europe. These are known breeding sites. So they've studied this before. And the temperature gradient is shown here. These little circles are where they hung out. So these are the different birds. So you might be wondering, how many did they put these backpacks on? There was only an N of 25 for this study, but it was still one of the really kind of first ones that followed them for so long. And again, there's like, this is kind of where they were, and this was their general, the circle shows you their general vicinity based on what the temperature was. Okay, let me make this a little bit smaller. And then I can show you the bottom one.
So far.
Okay, this is the comparison of temperatures between the breeding sites and the wintering sites during winter specifically. So wintering is the red here. Breeding is the blue temperatures. And then this shows you the temperature difference here. So maybe I do need to make it bigger. There. So they did this from 1979 to 2018. And basically, you can see that at the wintering site, so this red is the wintering sites, um, it tended to be here in January where they were breeding. So they went to like slightly warmer areas here. Here are the breeding sites in blue. Temperature was a little bit cooler. And then here's the temperature difference. I think I got that right. I need to wear my better glasses. Um, so just an example of tracking, long time frame of tracking, what you can get out of that kind of GPS data. And it's talking to the satellites, so they don't need to retrieve the bird, which is really helpful. And they can get it kind of live streaming and compare. All right, one thing you might want to think about doing as well is a quadrat method. So you could make one of these at home out of anything, really. I make them out of PVC pipe. But basically what this is it's a metric, it's an amount of space that you're going to measure things in. So a quadrat is a sampling air is basically something that a square that you create. You can tie sticks together as well. And you are defining it. It can be like half a meter by half a meter. It's kind of the easiest size to manage. And that what that means is 1/2 times 1/2 is a quarter metered squared. And what you do is if you have this big sampling area, let's say this is 20 feet by 20 feet, you want to collect data in your quadrat at random spaces. Lots of different ways to find random spots. Um, some people toss it out. Some people put transect lines out here and they look at a random number table and they say, okay, the first number is how far I'm going to go out this way, and the second number is how far I'm going to go out this way. So you can, like, if this is 15 feet, let's say, and you're always going to take like the first two numbers. Let's say it's 1, 6. You'll go out. Oh, that's too far because you've only done 16. So you go to your next number and let's say it's 6, 1. That's too many. Let's say the next one is 1, 3. Okay, so you go to the 13-foot mark here, and then your next number is 2. And you go out 2 feet. So you're right about here, and you put your quadrat down. In your quadrat, you can do multiple different things. You can count, let's say, the number of a certain type of plant, or you can count the percent cover of your whole quadrat. What makes a difference is what you're counting. So if you want to know, let's say, how much grass there is of a certain species, you might do percent cover because it'll be hard to like get down to the bottom and like decide like how many grass plants is this grass frond coming from. So you can do that. You can say, I'm not going to do like each plant because it'll take me forever. That's the way we do it with mussels, for example, in the intertidal. It's really, really hard. You cannot, it would take all day to like figure out, is there one mussel there, two mussels there? You could be there all day counting them. So what we do is percent cover. But for other things like sea stars, it's really easy to count how many there are in your quadrats. So quadrat method is just a way to collect abundance data in a certain space. It can be again tossed or it can be random numbers that generated. You just get a table and you just go through the numbers. And if it's again like 15 by 15 and you get a number that's too big, you go to the next number. Um, and you can pick and you can go out like if you can go out, and choose one digit that you're going to go out. The next one you could do choose two digits. If it's too much, you just use one. So this is a very, very basic but super informative way of collecting data. Here is just another example. So you measure an area and you form a grid, and then you take two random numbers and you use them as coordinates on your grid. And then this is what a quadrat looks like in the field. And then you count the number of species. You can even count the number of all the species. So this is something you could do as well for your mini study. It's not just one species, but you could decide, okay, in this grassland over here, I'm going to compare the species abundance to this grassland over here. Maybe you're just going to do it in this area. So you'll get a huge kind of plot and you'll randomly sample in there. It has to be random assignments of grids. And again, there's random number generators. The more quadrats you do, the better the validity of your study. Um, I would say for a study in this class, you could easily, depending on how much you're counting, do like 20 in a random area. So here's some key aspects of quadrat data collection. It is cost effective and reliable for populations of stationary organisms. So it's good for plants, slow moving creatures, mobile ones, or elusive ones could be missed. So you're not going to use quadrats to look at squirrels because they're not going to be sitting in your quadrat. So when you're analyzing the data, you can use statistical methods like ANOVA. It's used to analyze the data and interpret the relationship between the variables. So again, this is valuable for ecological studies, biodiversity monitoring, assessing the impacts of environmental factors like climate change. And you could do it somewhere near your house. So in 5 years, you can do it again and see what's changed. Another fun method is pitfall traps. So these are used to catch small ground-dwelling animals like beetles, spiders, and ants. So you have a container or a cup that's buried in the dirt with its rim flush with the ground, and then you usually want to have some sort of cover on the top. And that way, you're not also getting things raining down on the top. You're getting creatures. Let's say your goal is to study creatures that are moving through on the ground in this habitat. So you can put in the container some sort of liquid preservative like ethanol with some soap. You could also just put in like Dawn dishwashing soap to break the surface tension so that the insects, once they fall in, they can't escape. So it's passive collection. You can leave it for a few days. You can get a long-term sample of the community. It's not always, so this is the bias. None of these methods are perfect for everything. It's not always perfect indicator of abundance because activity levels differ between species. So certain things are going to move more than other things, and you're not going to see them because they haven't walked across. It's good for really active insects. They're most likely to be caught than less active ones. Another is sweep nets. Okay, so this is also fun. You see this big net. So this is a simple way of actively collecting insects in vegetation. So you basically walk through a vegetated area and you swing this canvas net back and forth in a wide arc and you sweep insects off of plants. You can do this for butterflies as well or moths, and you pick a line and you walk a certain distance. And it's important to kind of keep track of how long you walked and how your speed so that you're kind of doing the same thing in multiple different areas. So after you set up a lot of sweeps or a certain distance, you stop, you fold the net over, you trap the insects, and then you put them all in a jar or a Ziploc bag so that you can analyze them later. So this is really good for pest populations and agricultural fields. It's really good for general surveys of insects. It's good for butterflies, moths, flying little creatures. It's really cost effective. It gives you a good snapshot of the insects and the fauna at a specific time. You want to do it if you're going to do something like this and you're going to tell us about insect diversity. You might want to do one in the morning and one in the evening and compare what you see in your nets. This you'd need probably something that's harder about this is you'd need either a book or a good iNaturalist to help you identify. You might want to download like a microscopic camera onto your iPhone or your phone so that it can take a really close-up picture and that would help you identify what you're looking at, but that could be also really fun for you. Baited traps. Baited traps are just what they sound like. So it's a trap that has bait and you're trying to catch something. So we use this for trying to catch a skunk, let's say that's in someone's basement, maybe a raccoon that's infested, a garage. But it's also used by scientists to collect things. So, it's used to get bobcats sometimes, other types of cats, mountain lions. Those tend to not be a trap that they get into like this, though. It's more of um, like something that's a camera trap where they're just trying to see them and they'll put like a carcass out. Um, you can use different things depend in the trap depending on what you want to catch. So rotting fruit, dead animal dung, maybe a pheromone that's synthetic to attract a star target species. So it's placed either in or near the container. And then the hope is that it goes in and then something's triggered and it closes. So they're really effective for specific species. They're unique for what you're sampling. There's also something called sugaring that trees that they have fermented bait mixture, and it's used to trap nocturnal insects like moths. And creatures like this have a safe release. So that's what I like. They're not injured in baited traps. This is another cool thing. This is called a malaise trap. So it's basically a net at an angle out in an area, and it's really effective for collecting flying insects. A lot of wasps that are Hymenoptera in that family, really, really abundant. A lot of flies, dipteras, hit it, and it's basically it runs, they kind of get pushed up into the center, and then as they crawl upwards, to try to get out, because most of them do. They don't crawl down, they crawl up, and they're funneled to the peak of the tent that has a collection jar in it. So this white roof increases the light contrast, which helps guides the insects towards that collection bottle. You can leave these in place for a really long time. They're ideal for for getting like biological diversity of insects in an area. Placement is really important, so you have to kind of figure out a good place to put them, like forest edges or trails, to maximize what you're catching. And then here are just some examples of camera traps. So these are really non-invasive, which I like. They're automatic, usually placed on trees, to try to camouflage them in areas. And they're really valuable for observing behaviors like pollination, but also larger mammals that are hard to see and that are skittish around humans. There's verifiable video evidence that you can get. It's relatively inexpensive, and there's infrared lights on them, so it shows a lot of nocturnal activity, which is awesome. The data can be affected by the type of recording schedule and the motion sensor settings, so that can impact the detection of fast versus slow moving organisms. So that means if you have it on, it has to be like moving at a certain velocity to pick it up. Sometimes you miss things. There's also, if you have it constantly running, so much data to go through. So what most people use now is kind of targeted for a certain species size and movement speed, and then the camera will take a picture. They try to not use bright lights or any sort of flash because it scares the animal from using that space again. Um, but it's been really amazing to see data collected of like even our bears in behind Santa Barbara are being seen by a lot of camera traps. All right, mist netting. This is something I really do love to do. So this is when fine nets are deployed and it captures birds and bats. Um, and the birds kind of get their little feet caught in them, sometimes a wing. You run over, you gently take the bird out, you hold on to them really quickly. You can tag them, weigh them, measure their wings, their beak length, maybe their leg length, and then you release them, and off they go. Bird tags usually go on their legs. So this is really, really an effective way to kind of see where migratory routes are, what species are using this certain area. And what I like is I've never seen a bird get hurt in mist nets. So you are putting it up, they fly in, run over, get them off, take some measurements, potentially tag them. And this is how we know a lot of the routes and a lot of the distances birds are going. There are some opportunities at UCSB for doing field work, and one that I always like to talk about is the bioma that Wagner, one of our professors, runs in Costa Rica. I've had a lot of students do this over the summer, and I think you can choose a different number of weeks, but you basically are going out collecting data on turtles. And I've done a lot of turtle data collection. And you're kind of in, I believe in this program, you're in like a lab with other people, and you go out at night and you walk the beach and you check for females that are coming up and laying eggs, and you get to watch them actually digging their holes and laying their eggs. And you can use red light because that doesn't affect them and bother them. Once they start laying, they kind of go into a trance. You certainly don't go by their head or disrupt them in any way. But these programs have really, really helped. These monitoring programs our sea turtle population. They are these amazing organisms that use the entire ocean. We don't even know half of what they know in terms of where to go for food and where they swim. We just know that females always go back to the beach where they were born to lay their eggs. And so we need to keep those beaches predator free. And there's a lot of predators for sea turtles and their eggs. Everything from when they're in the sand, raccoons and small mammals kind of dig them up. Humans dig them up to eat the eggs. There's even a place where there's pumas, I think, that dig up. I can't remember what I saw that on. Um, and eat the eggs, and they also eat the turtles as they come up to lay eggs. So these turtles are protected by these students that do research there and these community groups that are committed. So it would be a really great experience if that's something you're interested in doing. I just love this picture of the Shumash rock art. So I like to show it. If you have questions, email me, go to your TA's office hours, and we want to hear from you about your ideas as well. And there's three more slides I'm going to put in that just have like more methods, descriptions, what they're used for, and examples, just for you to reference for your study. So these ones are a lot of the hands-on ones. We have more like direct counts, different sampling, indirect methods. That could be like counting scat or nests or tracks or burrows or vocalizations. So much fun stuff to do. There's demographic studies where you're looking at cohorts or survivorship curves. There's modeling. There's remote sensing and tech. There's an experimental manipulation. So I just want you to have all the different examples kind of at your fingertips in this lecture for when you're thinking about what you want to do. Thanks so much for listening. I'll see you soon.